Hello, my name is Vladimir, and I was in charge of doing the SDS page gel. First, I will explain the point of an SDS page. An SDS page helps us take fractions that were recommended by the FPLC and spectroscopy groups and identify which fractions are enriched in MGH. Something we don't want to see are fractions with lots of contaminants. To get a closer look at the fractions, we would perform a gel electrophoresis. And since we didn't physically create one in lab, a gel image, I'll be going over the steps that we would have done to obtain one. So we would have taken our samples and stained them with loading dye, heat them, centrifuge them, and load them into the wells of the gel. Along with the samples, we would load a marker and a pure MGH sample, and so the SDS will coat the proteins and make them negatively charged, which will separate them by the mass, and the marker will let us get an estimated size. And after running the gel, we would have stained them with Kamasi blue to see the proteins. So this is the gel image that was given to us, and I'll be going over it. So lane 1 includes the marker, which gives us an estimated size for the proteins. Lane 2 is the clear lysate, and the clear lysate lane includes the entire population of proteins subjected to FPLC purification. Lane 3 contains the flow-through, which is fraction 6, and this lane should contain a lot of contaminating protein, but not our protein. If our protein would be visible here, which it is because there's a band next to the big blob of MGH, then that means that we overloaded our column and our protein would come through because there wasn't a spot for the protein to sit on the resin. Lane 4 contains the authentic MGH protein and helps us identify MGH in all our other lanes. Lanes 5 to 10 contain fractions 19 to 24, and by the look of it, all contain MGH and no contaminants, meaning that the SPECT and FPLC teams did a good job with the recommendations. And we can tell that the MGH is in there because all of the bands look pure and look like they're good in yield next to the big blob that's in lane 4, which is the MGH. For future purification, if you would like to increase yield, you can increase the amount of cells you culture when you're overexpressing them, because if you have more cells, you'll have more MGH. To decrease degradation, you could increase protease inhibitors, since protease break down proteins. If we inhibit the breakdown of proteins, we prevent degradation. So all the fractions look good that were recommended to look over, and contain MGH with no contaminants and being high in yield. And I would take specifically fractions 20 and 23 because they seem slightly higher in yield. And I mean, they all look good, but 20 and 23 stand out slightly more to me.